You think you know the ocean. Blue water, some dolphins, maybe a shark if you're unlucky. But 300 meters down, the rules change. 600 meters and you're in a different world. 1,000 meters deep, and you've crossed into territory where pressure crushes submarines and darkness is absolute. Down there, fish have transparent heads. Males fuse their bodies to females and become living sperm banks. Eyes glow green in the blackness. Creatures eat garbage floating down from above because nothing else exists. This isn't science fiction. This is Earth, right now, under every ocean you've ever seen from a beach. The deep sea covers more than 60% of our planet's surface. We've explored less than 5% of it. In that tiny fraction, we've found creatures that rewrite everything we thought we knew about how life works. The blobfish that looks like melted wax when you pull it up. The vampire squid that's neither vampire nor squid. The anglerfish where females carry their mates fused into their bellies. Giant squid with eyes the size of dinner plates. And in January 2024, off the coast of Chile, scientists found over 100 new species in less than a month. 1 month, 100 species. We've been looking at the ocean for thousands of years and we're still finding things that don't make sense. So here's the real question. What else is down there? What's living in the 95% we haven't seen? And what happens when we finally drag it into the light and watch it fall apart because it was never meant to exist at surface pressure? If you value documented marine biology and deep sea research like this, subscribe. It's the best way to support our work. Also, comment from which city you're hearing this from. Also, this script is researched and factual, using sources from the Schmidt Ocean Institute's Falkor expeditions documented in their February 2024 publications on seamount biodiversity, the Monterey Bay Aquarium Research Institute's ROV footage analysis published in Proceedings of the Royal Society, and the Senckenberg Ocean Species Alliance's November 2025 Biodiversity Data Journal showing 14 newly described species using advanced taxonomic methods. These aren't Wikipedia summaries, these are primary research institutions with peer-reviewed data from expeditions that happened in the last 2 years. Now back to the information. Let's start with something that went viral for all the wrong reasons. The blobfish, you've seen the photos, pink blob sad face, voted ugliest animal on Earth in 2013. Cycrolutes marsidus, smooth head blobfish, lives off the coasts of Australia, Tasmania and New Zealand, 600 to 1,200 meters deep. That's 2,000 to 3,900 feet. At that depth, pressure is 60 to 120 times greater than at sea level. Your eardrums rupture at 7 feet deep. At 3,300 feet, your lungs collapse. The blobfish lives there every single day. Here's what nobody tells you. That melted, looking blob in all the photos. That's not what the fish looks like. That's what happens when you rip it out of its natural habitat, and the pressure differential destroys its body structure. Down in the deep, at 120 atmospheres of pressure, the blobfish looks normal. It looks like a fish, it has a proper face. The gelatinous flesh that makes it look pathetic at the surface is actually brilliant engineering. No swim bladder because gas bladders implode under that pressure. Instead, the flesh is a gelatinous mass with density slightly less than water. It floats above the seafloor without swimming, without effort. It just exists in the water column, mouth open, waiting for deep ocean crustaceans to drift by. No muscles needed, no energy wasted. It's adapted to an environment where food is so scarce that movement is a luxury it can't afford. The blobfish was scientifically classified in 1926, but the general public didn't know it existed until 2003. A research voyage off New Zealand hauled one up by accident. Dead, decompressed, destroyed. That corpse became the face of the species. Marine biologist Kerrion Parkinson found it during that expedition. Since then, every blobfish pulled from the deep arrives dead or dying. Bringing them to the surface is thought to be fatal, even with gentle handling. They're bycatch in deep sea trawlers, fishing for orange roughy and crustaceans. The nets sweep the ocean floor. The blobfish get caught, they surface, they die. Some scientists think the species is endangered. Others say we don't know enough about deep water populations to make that call. Either way, the blobfish has no natural predators except humans, and we're killing them by accident while looking for dinner. 30 centimeters long, 12 inches, whitish or light gray, lives in complete darkness. Most specimens we have are dead ones that washed up or got tangled in nets. The few images we have of living blobfish in their natural habitat came from remotely operated vehicles in the 2020s. They show a fish that looks almost boring. No dramatic features, just a regular deep sea fish doing regular deep sea fish things. Floating, waiting, existing. The pressure holds it together. Remove the pressure and you get the viral photo. It's like taking a human to the surface of Mars without a suit and then claiming that's what humans look like. Now go deeper, past where the blobfish lives, down to the oxygen minimum zone, 600 to 900 meters in some oceans, up to 3,000 meters in others. Dissolved oxygen drops to 0.4 milliliters per liter. Most marine life suffocates, but Vampiroteuthis infernalis lives there, the vampire squid from hell. Discovered during the Valdivia expedition between 1898 and 1899, German teuthologist Carl Chun found it and thought it was an octopus. It's not. It's not a squid either. It's the only surviving member of the order Vampiromorphida, a phylogenetic relic, something that should be extinct but isn't. 21 centimeters maximum mantle length, 8 inches, 8 arms, 2 retractile filaments that extend up to 8 times the animal's total length. The filaments aren't tentacles, they're something else, something unique to this species. Covered in sensory structures, they detect chemical signals and vibrations in the water. The vampire squid deploys them one at a time, drifts in the current, waits. When a filament contacts food, the squid investigates. It doesn't hunt, it scavenges. Detritus, marine snow, dead zooplankton, discarded larvation houses, crustacean remains, diatoms, fecal pellets from animals living in the waters above. It combines the waste with mucus secreted from its suckers, forms balls of food, eats garbage. This was discovered in 2012. For over a century, scientists thought vampire squid were predators, aggressive hunters in the dark. Then researchers at Monterey Bay Aquarium Research Institute used ROV footage and laboratory feeding experiments to prove otherwise. Hendrik Hoving and Bruce Robertson published their findings in Proceedings of the Royal Society. The vampire squid is a detritivore. It eats waste. It survives in a zone where oxygen levels would kill most cephalopods by having the lowest mass-specific metabolic rate of any cephalopod ever studied. Its blue blood's hemocyanin binds oxygen more efficiently than other species. Its gills have an especially large surface area, weak muscles, but sophisticated statocysts for balance. Ammonium-rich gelatinous tissue that matches the density of surrounding seawater. It doesn't fight the environment, it becomes part of it. Dark coloration, black chromatophores with reddish-brown ones interspersed. Off California, the reddish-brown dominates. Elsewhere, the black takes over. The name comes from that darkness and the webbing between its arms, like a vampire's cloak. But it doesn't suck blood. Carl Chun named it based on appearance, not behavior. The species name Infernalis, from hell. The genus Vampiroteuthis, vampire squid, a creature from nightmares that turns out to eat dead particles floating in the abyss. It has defenses. When threatened, it doesn't flee far because weak muscles limit stamina. Instead, it turns inside out. The pineapple posture. Arms and webbing spread over the head and mantle. Spiky structures called cirri stick out in all directions. If that doesn't work, it ejects a cloud of bioluminescent mucus from its arm tips. Blue glowing orbs, thousands of them. The display can last nearly 10 minutes. The light sticks to predators. The burglar alarm effect makes the attacker visible to secondary predators. Same strategy the Atolla jellyfish uses. The vampire squid then disappears into darkness without swimming far. The mucus regeneration costs energy, so it only uses this defense when extremely agitated. Large eyes and optic lobes adapted for detecting distant bioluminescence. Signs of prey aggregations. Potential mates. It monitors vast areas of the water column where most features don't exist. Found circumglobally in temperate and tropical oceans. Northern California's Monterey Bay, 600 to 900 meters deep. Oxygen concentrations around 0.4 milliliters per liter. The vampire squid's ability to thrive in oxygen minimum zones keeps it safe from apex predators that need more oxygen. It's found its niche, a place where almost nothing else can survive. And there it drifts, eating trash, glowing faintly, a living fossil from an ancient cephalopod lineage that split before octopuses and squids fully diverged. Giant grenadiers eat them, deep-diving mammals, whales and sea lions. Vampire squid show up in stomach contents, but those predators have to dive into the oxygen minimum zone and grab them fast. Most of the time, the vampire squid just floats there, filaments extended waiting for waste to drift down from above. It's sedentary. Generalist, it'll eat complete copepods, ostracods, amphipods, isopods, salp remains, larvation houses, medusa jellies, anything that falls from the zones above and makes it down to the suffocating depths where life moves slow and energy conservation is everything. The vampire squid uses a unique luring method. It agitates bioluminescent protists in the water, creates movement, light. That attracts larger prey, which it then consumes, but mostly it's passive. The vampire's longevity is unknown, reproduction poorly understood due to the difficulty of observing it. Marine biologists suggest that given their limited movement, pairs must simply cling to each other. Other Cycrolutidae species lay thousands of eggs. Researchers estimate a single female vampire squid could lay up to 100,000 eggs, but confirmation requires observation that hasn't happened yet. The deep won't give up its secrets easily. Now let's talk about the giant squid, Architeuthis dux, the kraken, the monster, the thing that shouldn't exist, but does. Largest invertebrate in the world, maybe, depending on how you measure against the colossal squid. Recent estimates put maximum body size at around 5 meters for females, 16 feet from posterior fins to the tip of the arms. Males slightly shorter. The mantle is about 2 meters long, 6 feet 7 inches, but the feeding tentacles concealed in life extend 10 meters, 33 feet. Total length 12 to 13 meters, 40 to 43 feet. Some claims say 20 meters or more, 66 feet. Those claims aren't scientifically documented. Every verified giant squid we've measured maxes out around 13 meters total length. The eyes, 10 inches in diameter, 25 centimeters. The size of a dinner plate, the size of a human head. Largest eyes in the animal kingdom. They share that distinction with the colossal squid. The eyes see in near total darkness. They detect the silhouettes of objects overhead using ambient light that barely exists. They distinguish bioluminescence from ambient light. They track small zooplankton with precision. The eyes are an evolutionary response to living between 300 and 1,000 meters deep, 980 to 3,280 feet. In the bathypelagic zone, the midnight zone, where less than 1% of surface light penetrates. The giant squid was always a legend until it wasn't. 1833, an almost perfectly spherical fish washed ashore in Greenland. Taken to Johannes Christopher Hagemann Reinhardt in Copenhagen. The football fish, Hymentolephus groenlandicus, the man gobbler. Wait, wrong creature. Let's get back to the squid. First image of a live mature giant squid was taken on January 15, 2002. Goshiki Beach, Amino-cho, Kyoto Prefecture, Japan. The animal measured about 2 meters in mantle length, 4 meters total. Found near the water's surface. Captured, tied to a key. Died overnight. Kutaru Tsuchiya of the Tokyo University of Fisheries identified it. It's on display at the National Science Museum of Japan. First footage of live larval giant squid was captured in 2001, shown on Chasing Giants. On the Trail of the Giant Squid on Discovery Channel. First photographs of an adult hunting in the wild came on September 27, 2005. Tsunemi Kubodera and Kyoichi Mori released them to the world. Photo sequence taken at 900 meters depth, 3,000 feet, off Japan's Ogasawara Islands. Shows the squid homing in on a baited line, enveloping it in a ball of tentacles. The squid impaled a tentacle on the bait ball's hooks. The researchers tracked sperm whales to find the general location. The whales fed on giant squid. The scientists followed the whales. The whales led them to the prey. Published in Proceedings of the Royal Society. Weight, 440 to 2,000 pounds, 200 to 907 kilograms, average around 455 kilograms or less, 1,000 pounds. Despite the massive size, they're not the heaviest cephalopods. The flesh is less dense than seawater. They maintain buoyancy with pockets of ammonia solution within the body walls. The fins are small. Muscles appear poorly developed. They're probably not fast swimmers. They ambush, they wait, they grab. Diet: deep sea fish, orange ruffy, hoki, other squid. Species: not other giant squid. Cannibalism isn't documented. Some suggest they might attack small whales. No evidence confirms that. The two long feeding tentacles shoot out, grip the prey. Suckers on the tips grab hold. Tentacles contract, bring prey to the arms. The arms subdue it further, pull it to the strong, sharp beak. The radula tears it apart, file-like teeth on the tongue. The prey is shredded before it reaches the esophagus. The giant squid has dark, sepia-colored ink, like smaller squid. Two large gills rest inside the mantle cavity. They breathe by expanding and contracting sets of muscles. Water fills the expanded space. Muscles relax. The elastic mantle snaps back. Jets water through the funnel. Locomotion and respiration in one system. They're found in all the world's oceans, North Atlantic especially, Newfoundland, Norway, Northern British Isles, Spain, Azores, Madeira, South Atlantic around Southern Africa, North Pacific around Japan, Southwestern Pacific around New Zealand and Australia, rare in tropical and polar latitudes. Genetic research published in March 2013 by the University of Copenhagen suggests there's only one species worldwide. Architeuthis ducks. Despite previous claims of multiple species, DNA analysis showed they're so closely related that they represent a single global population. No matter where they're found, they're the same species. Lifespan: 2 to 5 years. Some studies suggest they reach adult size within 3 years. Given rapid cephalopod growth rates, attaining 500 kilograms or more in fewer than 3 years is impressive but possible. Sexual maturity timing is unknown. Many recovered specimens have been mature, especially females. But we don't know when they reach that point or how reproduction actually happens in the wild. No two Architeuthis have ever been caught together in fishing nets. Scientists hypothesize they're solitary hunters. They hunt alone, live alone. Only meat to mate. The giant squid remains largely a mystery despite being the biggest invertebrate on Earth. Almost everything we know comes from carcasses that washed up on beaches or got hauled in by fishermen, dead specimens, decomposing bodies. The inhospitable deep-sea habitat makes them uniquely difficult to study. We've made progress. In 2004, researchers in Japan took the first images ever of a live giant squid. But even now, in 2025, we've only seen them a handful of times in their natural environment. Most of our data comes from dead animals. They're vertical. Distribution spans 300 to 1,000 meters, based on trawled specimens and sperm whale diving behavior. Sperm whales are their primary predator. The image of a giant squid locked in battle with a sperm whale is common in popular culture. But the squid is prey, not an equal combatant. The whale always wins. In the deep, the giant squid uses those massive eyes to see bioluminescence, to track prey, to avoid becoming prey. The eyes can distinguish shapes in the faintest ambient light. They have a high density of rod cells. No cone cells. Color vision doesn't exist at those depths. Everything is black or bioluminescent. The eyes adapt to that reality. 10 inches of light gathering power, scanning the abyss for anything that moves or glows. Recently, scientists aboard the Schmidt Ocean Institute's vessel Falkor made extraordinary discoveries. January 2024. They boarded the research vessel to explore underwater mountain ranges off Chile's coast, led by Dr. Javier Selanes of the Universidad Católica del Norte. They wanted to understand seamount biodiversity. They used remotely operated vehicles, multi-beam sonar, advanced sensors. In less than 1 month, they documented 100 newly discovered deep-sea animals, deep-sea corals, glass sponges, squat lobsters, Dumbo octopuses, siphonophores, jellies, an all-star lineup of the deep's most captivating residents, 100 new species. 1 month. These expeditions were part of a larger effort along the Salas y Gomez and Nazca ridges, underwater mountain chains in the southeastern Pacific, about 900 miles off Chile's coast, 1,450 kilometers. The newly found seamount rises 3,100 meters from the seafloor, nearly 2 miles tall. Its summit sits 994 meters below the sea surface. 600 meters is one depth estimate. Different seamounts, different depths. The region is exceptionally biodiverse. Before 2024, only about 1,000 species were documented in this Southeast Pacific region. Recent expeditions increased that number to more than 1,300. Dr. Erin Easton, lead scientist on some expeditions, said the findings blew them away. They were finding species they'd never encountered before every few meters. The creatures included cactus urchins. Only one known species exists currently: squat lobsters with spiky shells and beady eyes, ancient corals likely dating back thousands of years, translucent glass squids, potentially 30 new species of sea sponges, some unusually colorful, a bright red fish using its fins like hands to move across the seafloor. They witnessed it walking on the bottom, walking. Each seamount is its own ecosystem, distinct creatures that don't live anywhere else on the planet. Rotating currents carry cold water from the south and warm water from the north. High nutrient levels. These conditions create species highly specialized to thrive on the seamounts. Scientists are just beginning to unravel why species living in relative proximity on nearby ridges are so distinct. There's so much we don't know about these underwater biodiversity havens, but one thing is certain: what happens on these seamounts is directly connected to the larger ocean ecosystem. The expedition captured rare footage: a Casper octopus off the Nazca Ridge, a Promicoteuthis squid, a genus so rare that only 3 species have been described based on a few collected specimens, several from the late 1800s. They recorded it live, first time ever, a sea toad, Chornocops coloratus, at 1,430 meters depth, 0.9 miles. It clung to the newly discovered seamount, using its pectoral fins to brace against rock and sand. Despite its name, it's actually a fish. A scorpionfish, Helicolinus lengerici, appeared during survey of an unexplored seamount. Scientists haven't named the mountain yet. They just call it T06, the flying spaghetti monster, Bathyphysa conifera, a siphonophore, not an individual organism, a colony of tiny related individuals working together. They filmed it with its long noodle-like tentacles drifting through the water. Siphonophores can reach 30 feet in length. They're common in these depths. They trail thousands of stinging tentacles like living drift nets, capturing copepods and other small animals. Some siphonophores in the genus Apolemia grow to over 10 meters, 33 feet. Their tentacles stream out behind them, stinging anything they contact. Other creatures have learned to exploit this, to steal food right out of the tentacles. Which brings us to another bizarre adaptation: the barrel-eye fish, Macropinna microstoma, discovered in 1939 by marine biologist W.M. Chapman in deep temperate waters of the Pacific, Indian, and Atlantic Oceans. The presence of this fish was mostly unknown until 2004, when researchers at Monterey Bay Aquarium Research Institute photographed it off the North Pacific coast. Fossil evidence from a Russian study in 2016 indicates the genus Macropinna has existed for millions of years, traced back to the Miocene epoch. Similar tubular eye features preserved in a fossil species. The barrel-eye's distinct features aren't recent adaptations. They're ancient traits precisely evolved for deep sea life. The fish has a transparent head. Let me repeat that. A transparent fluid-filled shield covers the top of its head. You can see through it. Inside are 2 tubular eyes, bright green lenses. The eyes can rotate. Originally, scientists believed the eyes were fixed in place, only looking up. Bruce Robertson and Kim Reisenbichler proved otherwise in 2008, published in Copaya. The eyes can rotate both up and forward within the transparent dome. Maximum arc of rotation about 75 degrees. This allows Macropinna to observe prey while keeping its eyes protected from stinging tentacles of jellyfish, like organisms it feeds on. 6 inches long, 15 centimeters, lives around 600 to 800 meters depth, 2,000 to 2,600 feet, sometimes deeper, up to 1,015 meters in some reports. It has a tiny mouth. Most of its body is covered in large scales. The fish normally hangs nearly motionless in the water, using its large flat fins for stability and precise maneuvering. When it spots prey overhead, it pivots its body to bring the mouth up, while the eyes lock on target. The eyes rotate from dorsal to rostral relative to the body, or the fish tracks descending food, rotating its eyes to follow the path until the prey reaches mouth level. The green pigments in its eyes filter out sunlight coming directly from the sea surface. This helps the barrelly spot the bioluminescent glow of jellies or other animals directly overhead. When it spots prey, it rotates its eyes forward and swims upward in feeding mode. The transparent shield protects its delicate eyes from stinging cells. It's speculated that barrel eyes maneuver carefully among siphonophore tentacles, picking off captured organisms. The fish's eyes would rotate to keep eyes on the prize, while the shield prevents damage from nematocysts. At first glance, it seems the barrel eye's eyes are at the front of the head. 2 black holes, those aren't eyes. Those are olfactory organs, nares, analogous to human nostrils. The actual eyes are inside the transparent dome on top of the head, pointing upward most of the time. Until 2009, scientists had never successfully brought a barrel eye to the surface alive. The fragile, transparent shield was destroyed by pressure changes and rough nets. In 2009, Mbari researchers captured one alive, kept it in a shipboard aquarium for several hours, long enough to confirm the eyes rotate, long enough to understand how it actually lives. The barrel eye is thought to be a pelagic spawner. Eggs coated with oil float near the surface until they hatch. After hatching, larvae descend to the depths as they grow, feeding on zooplankton and small organic particles. No sexual dimorphism between males and females. No parental care. The fish just drifts in the abyss, eyes pointing up through its transparent skull, waiting for something to pass overhead. Its strong digestive system handles cnidarians and small drifting animals. The small marine creatures trapped on the tentacles of cnidarians are also targeted. It's a specialist thief, stealing meals from creatures that can't chase it. Protected by a transparent shield that evolved millions of years ago and hasn't changed because it works. In 2024, an Arctic deep expedition found more bizarre creatures. The Nippon Foundation Nekton Ocean Census Arctic Deep Expedition in May. Scientists studied samples in October at a species discovery workshop hosted by Ocean Census in collaboration with UIT. They found deep-sea anemones with bright red coloration. Red works as camouflage in the deep because red light is absorbed and scattered by water past a certain depth. Red objects appear black, blend into dark surroundings. Red-colored sea creatures become nearly invisible to predators. Arctic deep-sea creatures often have long arms, enhanced ability to capture food and navigate. In the dark, vast expanses, long arms reach and grasp prey or food particles that might be sparse and widely dispersed. Extended limbs sense surroundings, detect vibrations, chemical signals, movements in the water. Crucial for locating food and avoiding predators in pitch-black depths. Long arms also aid mobility, help creatures move across the seafloor or through the water more efficiently. Threadfin snailfish found at 2,461 meters at the Jotul vent field, thriving under extreme Arctic deep-sea conditions. These discoveries show how global collaboration and advanced imaging are transforming marine taxonomy, accelerating how species are documented and named. In November 2025, Senckenberg Ocean Species Alliance published findings in the Biodiversity Data Journal. 14 strange new species hidden in the deep sea. The parasitic isopod Zione everta has raised structures on the female's back that resemble popped corn kernels. Genus name derived from Zea, the corn genus, found in the Australian intertidal zone, represents a completely new genus. Researchers observed fascinating interactions among known deep-sea creatures. The tusk shell Laevidentalium visi collected from depths exceeding 5,000 meters, with a sea anemone attached to its anterior side. a relationship never before documented within this genus. Earth's oceans hold extraordinary biodiversity. Only a small portion of an estimated 2 million marine species have been formally identified and described. One of the biggest hurdles in ocean science is the lengthy delay between when a species is first discovered and when it's officially documented, sometimes decades. To overcome this bottleneck, researchers launched Ocean Species Discoveries, a data-rich publication platform designed for concise, high-quality marine invertebrate species descriptions. Faster standardized system dramatically shortens publication time. Speed is essential. Many marine species face extinction due to human-driven biodiversity loss before scientists learn they exist. Now let's discuss something truly disturbing: the anglerfish. Over 170 known species in 12 families of deep-sea anglerfish. Huge diversity within those families. They sport a fantastic range of shapes and textures. Some are squat and round, like Melanocetus johnsoni. Others are flat and huge, snouted like Thaumatictis binghami. Some are covered in whiskery filaments like Caulifrine jordani. Common names hint at wild forms: snaggletooth sea devil, wolftrap, pugnacious dreamer, tyrannical toad. They all share one feature: a bioluminescent lure extending from their heads. The esker, the fishing rod, the light comes from bacteria, symbiotic bacteria living in the lure. The bacteria are acquired from the environment. In 2019, Baker, Fried, Eason, and others published research in eLife showing that diverse deep-sea anglerfishes share a genetically reduced luminous symbiont acquired from the environment. The bacteria colonize the esker, produce light through chemical reactions. The anglerfish uses this light to attract prey in total darkness. The anglerfish was first known to science when an almost perfectly spherical fish washed ashore in Greenland in 1833. Taken to Johannes Christopher Hagemann Reinhardt in Copenhagen, Denmark, the football fish, Haemontolophus groenlandicus, the man gobbler. That was the first anglerfish known to science. Theodore Peach wrote about it. Oceanic anglerfishes, extraordinary diversity in the deep sea, University of California Press, 2009. Since then, about 170 species have been identified in 12 families. Female anglerfish have the lure. Males don't. Males are much smaller, in some species 10 times smaller. Sexual dimorphism taken to an extreme. Why? Because of sexual parasitism, the most bizarre reproductive strategy in the animal kingdom. When a male finds a female, he bites into her skin, releases an enzyme. The enzyme breaks down tissues at the point of contact. Over time, the male's circulatory system fuses with the female's. He becomes permanently attached. He loses his eyes, loses his fins, loses his internal organs, retains only his testes. He becomes a sperm-producing appendage, a living sperm bank attached to the female's body. When she's ready to spawn, he's already there, permanently, forever. This isn't speculation. We have specimens, females with multiple males fused to their bodies. One study found a female with 4 parasitic males attached. 4. In 2020, research published in Science by Jeremy Swan and others explained how this is possible. Anglerfishes lack genes to produce most molecules that would attack foreign tissue. Few or no T cells. Few or no antibodies. No functional adaptive immune system. This lack of immune response would kill most vertebrates. But it's exactly what anglerfish need for sexual parasitism. Dr. Thomas Boehm, study co-author, said this lack of immune system would kill a human. But for anglerfish, it's essential. The attachment is similar to organ transplantation. The males become part of the female's body. They share a circulatory system. The female provides nutrients. The male provides sperm on demand. This adaptation makes sense in the deep sea, where encounters between individuals are extremely rare. Finding a mate in the vast darkness is difficult. Finding the same mate repeatedly is impossible. So when a male finds a female, he doesn't let go. He physically fuses, becomes part of her, ensures reproductive success. However, sexual parasitism is surprisingly confined to few taxa. Despite more than a seven-fold increase in known parasitized females in collections throughout the world in the past 50 years, permanently attached males have been found in only 5 of the 11 ceratioid families, 10 of the 35 genera, 23 of the 160 recognized species. Theodore Peach published comprehensive research on this in Ichthyological Research in 2005. Evidence reaffirms 3 reproductive modes: obligatory parasitism, facultative parasitism, temporary non-parasitic attachment. Sexual parasitism has evolved separately at least 3 and perhaps 5 or more times within the suborder. It's not universal. Some anglerfish species have larger males that don't attach parasitically, but the species that do have parasitic males represent some of the most extreme adaptations to deep sea life. The bioluminescent lure plays a crucial role. Only females have it. The lure hangs in front of the mouth, glows in the darkness. Small fish and crustaceans see light, move toward it. The anglerfish's huge mouth opens, rows of sharp teeth snap shut. The prey is swallowed whole. The lure might also help males find females. In the pitch-black deep, bioluminescence is the only light. A male anglerfish has highly sensitive olfactory organs. He can detect the female's scent. He can see her glowing lure. He follows the light sometimes for miles across the vast emptiness. When he finds her, he bites, releases enzymes, begins fusion. His body dissolves until only reproductive organs remain. Anglerfishes live at depths of 200 meters or more, 650 feet, down into the Bath hypelagic zone, 1,000 to 4,000 meters, 3,300 to 13,000 feet. The midnight zone. No sunlight penetrates, temperature near freezing, pressure extreme, food scarce. In this environment, every adaptation is about survival. The lure attracts prey. The huge mouth captures prey. The expandable stomach stores prey. The parasitic male ensures reproduction. The lack of immune system enables fusion. Everything works together. The anglerfish can't survive in shallow water. Highly adapted to deep sea conditions. High pressure, low temperature, complete darkness. If brought to the surface, they die quickly. Their bodies are built for depth. Soft bones, reduced density, pressure-resistant physiology. Surface conditions destroy them. We've only seen a handful of live anglerfish in their natural habitat. Most of our knowledge comes from specimens caught in nets. Dead specimens. Preserved specimens. Deep water trawls bring them up occasionally. Always dead. The pressure change kills them. We study the corpses and try to understand how they lived. If you value documented marine research like this, subscribe. It's the best way to support this work. Also comment from which city you're hearing this from. This research is factual, drawing from peer-reviewed sources including Robison and Reisenbichler's 2008 publication in Copaea on barrel eye rotation, confirmed through live specimen observation at Mbari, Hoving and Robison's 2012 Proceedings of the Royal Society Bee Study, definitively proving vampire. Squid are detritivores through ROV footage and feeding trials, and the Ocean Census Arctic Deep Expedition data, published by the Nippon Foundation Nekton collaboration in 2024 documenting anemones, snailfish, and isopods at depths exceeding 2,400 meters, with novel morphological adaptations. These aren't nature documentaries. These are primary research papers with methodology sections and statistical analysis. Now back to the information. The deep sea is full of these adaptations, creatures that look impossible. Fish with transparent heads, squid that eat garbage, males that fuse to females, but they're not impossible. They're real, they're documented, they're living right. Now in waters we can barely reach, and we keep finding more. 20 new species were identified in a recently discovered underwater ecosystem. August 2024. At least 20. Some reports say more. Schmidt Ocean Institute expeditions along the Nazca Ridge, 900 miles off Chile's coast. Jotica Virmani, executive director, told ABC News the modern technology allows deep water exploration more accurately than ever before. Remotely operated vehicle named Sebastian collected images and samples. much greater accuracy than satellite imagery used in the past to map the seafloor. New species discovered include anemones, urchins, corals, shrimp, squat lobster. Fossilized whale bones may lead to discovery of a new species of ancient whale. First ever recording of the rare enigmatic Promachoteuthis squid. The pristine ecosystem is so far from any coastline, it appears almost untouched by pollution. Currently, about 26% of the ocean floor has been mapped to high resolution. We're working toward a full map of the seafloor. That would be the first time in human history we know what our planet's shape actually is. The findings are incredibly important for understanding ocean life in the Bath Pelagic Zone, between 1,000 and 4,000 meters deep, the largest animal habitat on Earth. We know less about it than the surface of the Moon. We've sent more people to space than to the deepest parts of our own ocean. In 1960, Jacques Picard and Don Walsh descended to the Challenger Deep, 10,916 meters, 35,814 feet, the deepest known point in the ocean. They saw a flatfish and shrimp. Life exists even there. We've been back a handful of times since. Every time we go, we find something new. The Mariana Trench, 11,000 meters deep at its deepest point, 36,000 feet. Pressure is 1,000 times greater than at sea level, 1,000 atmospheres. Steel submarines implode at those depths, but creatures live there. Snailfish, amphipods, sea cucumbers, bacteria. They're adapted to conditions that would kill nearly everything else on Earth. Their cell membranes contain different lipids. Their proteins fold differently. Their metabolisms run slower. They don't fight the pressure, they work with it. In 1977, scientists discovered hydrothermal vents, underwater hot springs on the ocean floor where tectonic plates spread apart. Water heated by magma erupts through cracks. Superheated water. 380 degrees Celsius, 716 Fahrenheit. But it doesn't boil because of the pressure. The water is packed with minerals, chemicals, hydrogen sulfide, methane, toxic to most life. But around these vents, entire ecosystems thrive. Tube worms, 2 meters long, no mouth, no digestive system. They have bacteria in their tissues, symbiotic bacteria. The bacteria use hydrogen sulfide to produce energy, chemosynthesis. The tube worms get nutrients from the bacteria. The bacteria get hydrogen sulfide from the worms. It's a system completely independent of sunlight, life that doesn't need photosynthesis, life that runs on volcanic chemicals, giant clams, yeti crabs, vent octopuses, scale worms, vent fish. They all cluster around these hotspots in the freezing deep. The vents create oases, islands of life in the desert of the abyss. When a vent goes extinct, the ecosystem dies. The creatures can't survive without the heat and chemicals, but new vents open elsewhere. The creatures disperse as larvae, drift in currents, find new vents, colonize them. The deep sea floor is dotted with these chemosynthetic communities. We found them in every ocean, Atlantic, Pacific, Indian, Arctic. Wherever tectonic activity creates vents, life appears. Cold seeps are similar but different. Methane and hydrogen sulfide seep up through sediment, slower than vents, colder, but still packed with chemicals, still supporting ecosystems, clams, mussels, tube worms, bacterial mats, all using chemosynthesis. Some cold seeps have been active for thousands of years. The communities are ancient, stable, slow-growing. A clam might live for a century, growing millimeters per year. When humans disturb these areas, recovery takes centuries. The deep sea is also filled with marine snow, organic particles drifting down from the surface, dead plankton, fecal pellets, molted shells, mucus. It falls constantly, a slow rain of detritus. This is the base of the deep sea food web. Everything that doesn't live near hydrothermal vents or cold seeps relies on marine snow. The vampire squid eats it directly. Sea cucumbers eat it. Brittle stars, sea urchins. They crawl across the seafloor, hoovering up particles. Creatures living in the water column filter it, extend arms or tentacles to catch falling debris. It's not a rich food source. It's low quality, low energy, but it's all there is. so life adapts. Metabolism's slow, growth rates decrease, reproduction becomes infrequent. Everything moves in slow motion. A sea cucumber might wait weeks for a good patch of marine snow. A giant isopod might go months without eating, but they survive because they've learned to exist on almost nothing. Giant isopods, Bathynomus giganteus, up to 76 centimeters long, 30 inches, 2.3 kilograms, 5 pounds. They look like pill bugs, roly-polies, but huge. They're crustaceans, related to shrimp and crabs, found at depths of 170 to 2,130 meters, 550 to 7,000 feet. They scavenge. Dead whales, dead fish, dead squid. Anything that falls to the bottom. They swarm corpses, tear into flesh with powerful mandibles. They can survive for years without food. Their metabolism is extremely slow. In captivity, giant isopods have gone 5 years without eating. 5 years. Then they eat once and go dormant again. In the wild, food falls rarely. When it does, they gorge, then wait. Japanese spider crabs, Macrochira caempheri, leg span up to 3.8 meters, 12.5 feet, largest arthropod by leg span, found in waters around Japan, depths of 50 to 600 meters, 160 to 2,000 feet. They're omnivorous, scavengers, eat dead animals, plants, algae, shellfish. They walk slowly across the seafloor, on spindly legs. They can live up to 100 years. Growth is slow. They molt as they grow. Each molt is risky. The crab is vulnerable. Soft-bodied, predators can kill them easily, but most survive. Keep growing, keep molting, keep walking. Dumbo octopuses, Grimpoteuthis, named for the ear-like fins that resemble Dumbo the elephant. Depths of 3,000 to 4,000 meters, 9,800 to 13,000 feet. Some found as deep as 7,000 meters. 23,000 feet, they're small, 20 to 30 centimeters long, 8 to 12 inches. They hover above the seafloor, flap their fins, pulse their arms, move gracefully through the water. They eat snails, bivalves, crustaceans, worms. They swallow prey whole, no venom, no beak crushing, just engulf and digest. Different species live at different depths. Some are red, some are pink, some are white. We've only seen them a few times. They're difficult to find, difficult to study. Most of what we know comes from accidental captures in deep sea trawls, gulper eels, Uropharynx pelecanoids, huge mouths, jaws that unhinge, stomach that expands. They can swallow prey larger than themselves, found at depths of 500 to 3,000 meters, 1,600 to 10,000 feet, length up to 60 to 75 centimeters, 24 to 30 inches. Most of that length is tail, thin whip-like tail with a light-producing organ at the end. Bioluminescence. They use the light to attract prey. The huge mouth scoops up fish, crustaceans, cephalopods. They probably don't swim actively. They probably drift. Wait. Ambush. The deep sea is full of ambush predators. Energy conservation is key. Don't chase food, wait for food to come to you. Frilled sharks, Chlamydocelicus anguineus, primitive sharks. They look like eels. Long, slender bodies, 6 pairs of gills with frilled edges, mouth full of needle-like teeth. 300 rows, they eat squid, fish, other sharks. Found at depths of 120 to 1,500 meters, 400 to 5,000 feet, length up to 2 meters, 6.6 feet. They're ovoviviparous. Eggs hatch inside the female. She gives birth to live young. Gestation period is 3.5 years, longest of any vertebrate. The young develop slowly, born fully formed, ready to hunt. Frilled sharks are rarely seen. They're deep dwellers, occasionally caught in nets, occasionally wash ashore. Each sighting is documented, each specimen studied. We're still learning basic biology. Goblin sharks, Mitsukurina ostoni, pink skin, long flattened snout, jaws that extend forward, depths of 270 to 1,300 meters, 880 to 4,260 feet, length up to 3.8 meters, 12.5 feet. They use electroreception, ampullae of Lorenzini, detect electric fields produced by prey. The snout is packed with these sensors. They swing their head back and forth, scan the seafloor. When they detect prey, the jaws shoot forward. Projectile jaws snap shut, retract. The prey is trapped. The goblin shark is a living fossil. The family Mitsukurinidae dates back 125 million years. One species remains, the goblin shark, unchanged, surviving in the deep, doing what it's always done. Cookie-cutter sharks, Icystius brasiliensis, small sharks, 42 to 56 centimeters, 16 to 22 inches, found at depths of 85 to 3,700 meters, 280 to 12,000 feet. They're parasitic, sort of. They attach to large fish, whales, dolphins, bite out a chunk of flesh, cookie-shaped wound. The teeth are designed for this, small sharp teeth in the upper jaw, large triangular teeth in the lower jaw. They latch on, rotate, remove a plug of tissue, then release, swim away. The victim survives usually, heals eventually. Cookie-cutter sharks have been found with chunks taken out of submarines, rubber sonar domes bitten, cables damaged. They'll bite anything soft. Six-gill sharks, Hexanchus griseus, large sharks, up to 4.8 meters, 15.7 feet, 6 gill slits instead of the usual 5, found at depths of 90 to 2,500 meters, 300 to 8,200 feet. They're opportunistic, eat fish, rays, seals, other sharks. Squid, crustaceans, carrion. They hunt at night, move to shallower water after dark, return to the deep at dawn. This daily vertical migration is common in the deep sea. Follow the food. Food moves up at night to feed in surface waters. Predators follow. At dawn, everyone returns to the depths. The ocean has layers. Epipelagic zone, surface to 200 meters, where sunlight penetrates, where photosynthesis happens. Mesopelagic zone, 200 to 1,000 meters, twilight zone, dim light, no photosynthesis. Bathypelagic zone, 1,000 to 4,000 meters, midnight zone, no light. Abyssopelagic zone, 4,000 to 6,000 meters, abyssal zone, near freezing. Immense pressure, hadal-pelagic zone, 6,000 to 11,000 meters. Hadal zone, ocean trenches, deepest parts. Each zone has distinct life, different adaptations, different strategies. The vertical migrators connect these zones. Lanternfish, Myctophidae, small fish, 2 to 30 centimeters, 0.8 to 12 inches, bioluminescent photophores on their bodies. They live in the mesopelagic zone during the day. At night, they rise to the epipelagic zone, feed on plankton, return at dawn. This is the largest daily migration on Earth by biomass. Billions of lanternfish, rising at sunset, descending at sunrise, every single day. They're eaten by larger fish. Squid, marine mammals. They're a crucial link in the ocean food web, transferring energy from surface to depth. Hatchetfish, Sternoptychidae, another bioluminescent migrator, flat bodies, reflective scales, photophores on their bellies. Counter-illumination. When viewed from below, the light from their photophores matches the dim light from above. They blend in. Predators can't see their silhouette. Depths of 200 to 600 meters during day. Shallower at night. Small fish, up to 12 centimeters. They eat copepods. Small crustaceans, larvae. They're eaten by tuna. Dolphin fish, larger predators. The cycle continues. Viperfish, Charliodus, long fangs, huge, mouth, bioluminescent photophores, depths of 200 to 5,000 meters. Vertical migrators, hunt at night in shallower waters, rest at depth during day. Length up to 35 centimeters, 14 inches. They eat fish, crustaceans, anything they can catch. The fangs are so long they don't fit inside the mouth. The viperfish can't close its mouth completely. It impales prey on the fangs, swallows it. The stomach expands. They can eat prey half their size. In the deep, when food appears, you eat it, no matter how big it is. Dragonfish, Stomiidae, similar lifestyle. Bioluminescence, long fangs, extendable jaws. Some have a light organ on a barbel under the chin. Red bioluminescence in some species. Most deep-sea creatures can't see red light. The dragonfish can. It shines red light, sees prey that can't see the light. An invisible spotlight. Other dragonfish have photophores along their bodies. Counter-illumination. Camouflage. They hunt at night, rest at depth during day. Vertical migrants, part of the daily cycle. The deep sea also has permanent residents, creatures that never leave the depths. Tripod fish, bathypteroids. They stand on the seafloor. 3 elongated fin rays, 2 pelvic fins, 1 caudal fin, like a tripod. They face into the current, wait for food to drift by, extend their pectoral fins. The fins have ray-like projections, sensory structures, detect vibrations, water movement, prey. When something passes, they snap it up. They can stand for hours, days, barely moving. Energy conservation. Depths of 200 to 6,000 meters, widespread. Multiple species, each adapted to standing still and waiting. Sea pigs, scotoplanes, sea cucumbers. They crawl across the abyssal seafloor. Tube feet, tentacles around the mouth. They eat organic particles in the sediment. Mud, marine snow, bacteria. They're detrivores, found at depths of 1,000 to 5,000 meters, lengths of 10 to 15 centimeters, 4 to 6 inches. They travel in herds, groups of sea pigs moving together, following food concentrations. When whale falls occur, sea pigs congregate, hundreds of them, feeding on the carcass. Whale falls are major events in the deep. A dead whale sinking creates an ecosystem. Lasts for years. Whale falls. When a whale dies and sinks, it brings tons of organic matter to the deep, 30 to 40 tons for a large whale. The carcass is consumed in stages. First stage. Mobile scavengers arrive. Hagfish, sleeper sharks, giant isopods, amphipods. They strip the flesh. This stage lasts months. Second stage. Enrichment opportunist stage. Polychaete worms colonize the bones. Crustaceans, mollusks. They feed on organic particles. This stage lasts years. Third stage, sulfophilic stage. Bacteria break down lipids in the bones, produce hydrogen sulfide. Chemosynthetic bacteria use the sulfide. Tube worms arrive, clams, mussels. The whale bones become a cold seep. This stage lasts decades. Fourth stage, reef stage. The bones become hard substrate. Corals settle, sponges, anemones. The skeleton becomes a reef, lasts for centuries. Over 100 species have been found on whale falls, some found nowhere else. Osidax worms, bone-eating worms. They bore into whale bones, digest the lipids. They have no mouth, no gut. Bacteria in their tissues do the digestion. Symbiotic bacteria. The worms are found only on whale falls, dependent on dead whales. Males are microscopic, live inside the females, hundreds of males per female. Sexual dimorphism extreme. Whale falls are stepping stones. They allow deep-sea species to disperse. Larvae drift from one whale fall to another, colonize, reproduce, repeat. Without whale falls, some species couldn't exist. The deep sea is hostile, but life persists. Adaptations are extreme. Bioluminescence, counter-illumination, transparent heads, tubular eyes, gelatinous bodies, low metabolisms, slow growth, long lifespans, delayed reproduction, small brood sizes, large eggs, parasitic males, symbiotic bacteria, chemosynthesis, scavenging, filter feeding, deposit feeding, predation, parasitism. Every strategy that works on land works in the deep. Modified, refined, pushed to limits. But the deep is under threat. Deep sea mining. Companies want to extract minerals from the seafloor. Manganese nodules, polymetallic sulfides, cobalt crusts, rich in rare earth elements, valuable. The mining would destroy the seafloor, scrape away the surface, kill everything. The ecosystems take centuries to recover, maybe longer. We don't know. We haven't studied it enough. We haven't mapped most of the ocean floor. We haven't identified most of the species living there, but mining companies are pushing forward, claiming they need the minerals, claiming it's sustainable. Scientists disagree. They say we should map the ocean first, identify the species, understand the ecosystems, then decide if mining is worth the cost. Deep-sea fishing is another threat. Bottom trawling, dragging weighted nets across the seafloor, catching everything. Orange roughy, deep-sea fish, slow-growing, long-lived, low reproduction rates, populations collapse quickly, can't recover. Bycatch includes corals, sponges, all the habitat-forming species. Seamounts are particularly vulnerable. They're biodiversity hotspots, each one unique. Trawling destroys them, flattens them, removes the structure. The species that lived there have nowhere to go. Some seamounts have been trawled so heavily that the ecosystem is gone, permanently. Climate change affects the deep too. Ocean warming. Surface waters warm. Deep waters warm slower, but they warm. Currents change. Oxygen levels drop. Ocean acidification. Dissolved carbon dioxide forms carbonic acid. pH drops. Shells dissolve. Corals bleach. Even in the deep, the oxygen minimum zones are expanding. Lower oxygen, larger areas, creatures that can tolerate low oxygen spread, creatures that need more oxygen retreat. Ecosystems shift. We're watching it happen. We don't know the full consequences. We won't know for decades. Plastic pollution reaches the deep. Microplastics, tiny particles, they sink, accumulate in sediment. Deep sea creatures ingest them, mistaking them for food. The plastics block digestive tracts, leach chemicals. We've found plastic in the Mariana Trench, the deepest place on Earth. We've found it in amphipods from the Hadal Zone. We've found it everywhere we've looked. The ocean is filling with our waste, even the places we can't reach, but there's hope. Marine protected areas, no fishing zones, no mining. Let the ecosystems recover. The Salas y Gomez and Nazca Ridges. Negotiations to create a blue corridor, protect the seamounts, protect the new species. The United Nations finalized the High Seas Treaty, protecting international waters, setting rules for marine protected areas. Challenges remain: formal adoption, ratification by at least 60 countries, implementation, enforcement. But it's a start. Scientists continue to explore, to document, to discover. Every expedition finds something new. Every dive reveals species we've never seen. The ocean is vast, deep, full of life we're just beginning to understand. The blobfish that isn't actually ugly, the vampire squid that eats waste, the anglerfish with parasitic males, the barreleye with transparent head, the giant squid with dinnerplate eyes. These aren't movie monsters. They're real, living, thriving, in conditions that would kill us instantly. And we keep finding more. In 2025, scientists will continue mapping seamounts, deploying ROVs, collecting samples, analyzing DNA, describing species, publishing papers. The work never stops. Each discovery raises new questions. How did this evolve? Why does it look like that? What does it eat? How does it reproduce? How long does it live? The questions multiply faster than the answers. The deep sea remains Earth's final frontier. We've been to the moon. We've sent rovers to Mars. We've photographed distant galaxies. But we haven't explored most of our own ocean. The Mariana Trench has been visited fewer times than the summit of Mount Everest. Deep sea research is expensive, difficult, dangerous. The ROVs cost millions. The research vessels cost more. The missions require planning, funding, expertise, years of work for hours of footage. But every hour reveals something incredible. Recently, scientists found a fish that walks on its fins, bright red, uses pectoral fins like hands, moves across the seafloor, step by step. It's not supposed to be possible. Fish don't walk, but this one does. Filmed during a seamount survey. Another species that shouldn't exist, but does. Another adaptation to an environment we barely understand. Cactus urchins. Only one known species existed. Now they've found more, different colors, different spine patterns. Each seamount has unique urchins. Why? What evolutionary pressure creates that diversity in such close proximity? We don't know yet. We're still collecting samples, still sequencing DNA, still trying to understand. Glass sponges, hexactinellida, built from silica, glass-like skeletons, found at depths of 200 to 6,000 meters. They can live for thousands of years. Individual sponges carbon dated to 15,000 years old. The oldest animals on Earth. They filter feed, pass water through their bodies, extract organic particles, bacteria. They're sessile, rooted to the seafloor, can't move, just exist, filtering, growing, slowly, millimeters per decade. They form sponge gardens, entire fields of glass sponges, provide habitat for other species, fish, crustaceans, echinoderms. Remove a sponge garden and the community collapses. The Seamount expeditions found pristine sponge gardens, 800 square meters, twice as long as a basketball court, undamaged, untouched. Scientists hope to keep them that way. Deep sea, corals, scleractinia, cold water corals, no zooxanthellae, no photosynthesis. They feed on plankton. Organic particles, grow slowly. Some colonies are thousands of years old. They form reefs, structure, habitat, like their shallow water cousins, but in total darkness, in near freezing water, under immense pressure. They've been growing for millennia. When trawlers destroy them, they don't grow back. Not in human lifetimes, maybe not ever. Chrysogorgia, golden corals, bright yellow, fan-shaped, found on seamounts, depths of 200 to 5,000 meters. They host communities, squat lobsters, ctenophores, sea stars. The corals provide structure, protection, feeding opportunities. Remove the coral and the community disappears. Seamount biodiversity depends on these habitat formers. Protect the corals, protect the sponges, protect everything else. The expeditions also filmed predators, king crabs, neolithodes, massive, spiky shells, claws. They walk slowly across the seamount slopes, hunt, scavenge. They're related to hermit crabs, not true crabs, asymmetrical abdomens. They've been around for millions of years. Fossils show similar forms in the Cretaceous. 100 million years ago, they've barely changed. Why change what works? Brittle stars, Ophiuroidea, 5 arms, flexible. They crawl across the seafloor, wave their arms, filter feed, catch particles. They can regenerate lost arms, break off an arm and it grows back. Autotomy, self-amputation, defense mechanism. Predator grabs an arm, the brittle star drops it, escapes, grows a new one. Found from shallow water to the Hadal zone, everywhere. One of the most successful groups in the ocean. Sea anemones, actiniaria, cnidarians, tentacles, stinging cells, nematocysts. They attach to rocks, corals, shells, anything hard. Wait for prey to brush the tentacles. Sting, paralyze, pull to mouth, digest. Some species form partnerships. Clownfish and anemones. The fish provides food scraps. The anemone provides protection. Mutualism. In the deep, anemones are often bright red. Orange-white, colors we don't expect, we don't know why. No light to see the colors. Maybe chemical signaling. Maybe leftover from shallow water ancestors. Maybe we're wrong about what colors mean in the deep. Feather stars, crinoidea. Look like plants. They're animals. Echinoderms, related to sea stars and sea urchins. Feathery arms. 10, 20, sometimes more. They attach to substrate, wave their arms in the current, filter feed, catch plankton, organic particles. They can swim by flapping their arms. Moving from one spot to another, hunting for better currents, better food. Found from shallow water to 8,000 meters. Hardy, successful, ancient. Fossils date back to the Ordovician, 480 million years ago. They survived 5 mass extinctions. Whatever we do to the ocean, crinoids will probably survive. The deep sea is full of survivors, specialists, ancient lineages, living fossils. They've found strategies that work. Stick with them. Don't change. Don't evolve unless necessary. Energy efficiency, slow metabolism, long lifespan, low reproduction. These traits appear again and again. They're the key to deep-sea success, but they're also the reason deep-sea species are vulnerable. Long lifespans mean late maturity. Late maturity means slow population growth. Kill, too. Many adults and the population crashes. Recovery takes decades, centuries, maybe never. Orange roughy can live 150 years. They don't reproduce until age 20 to 30. Fishing pressure collapsed multiple populations. Some haven't recovered 30 years later. Might never recover, we don't know. Deep-sea species are K-selected. Few offspring, high parental investment, slow growth, long life, low mortality. This works in stable environments. The deep is stable. Temperature doesn't fluctuate. Salinity is constant. Pressure is constant. Food availability is low but predictable. Marine snow falls continuously. Whale falls arrive occasionally. Predictably. Life adapts to stability. But stability makes them vulnerable to change. Sudden change, human-caused change, fishing, mining, warming, acidification, pollution. The deep isn't adapted to rapid change. Species can't respond fast enough. Shallow water species are often R-selected. Many offspring, low parental investment, fast growth, short life, high mortality. This works in unstable environments, temperature swings, storm disturbance, predation pressure. Species adapt to chaos, reproduce quickly, die quickly, populations recover quickly. Fishing pressure on shallow species can be sustained if managed properly. Populations bounce back. Cod, herring, sardines. Overfishing causes crashes, but recovery is possible. The deep doesn't work that way. Overfish deep. Species and they disappear. Orange roughy, grenadiers, deep sea sharks. We're learning this the hard way. We're destroying fisheries before we understand them. The solution is obvious. Stop fishing the deep. Stop mining the deep. Map the ocean, study the species, understand the ecosystems, then decide what's sustainable. But economics drives policy. Companies want profits now, not later. Not after we understand things. Now. Politicians respond to companies, not scientists. Money talks. Conservation doesn't. Unless we make it, unless we demand it. Vote for ocean protection. Support marine reserves. Reduce carbon emissions. Reduce plastic use. Pressure companies. Pressure governments. Make. Noise. The ocean has no voice. We have to be its voice, and we have to hurry. The ocean is changing fast. Warming, acidifying, losing oxygen. Species are moving, shifting ranges, following temperature, following oxygen, following food, but some can't move. Endemic species. Species found on one seamount, one vent, one seep. They can't move to another seamount, can't cross the abyssal plains, can't find similar habitat. Destroy their home and they're extinct. Gone. Forever. We're causing extinctions we'll never know about. Species going extinct before we discover them, before we name them, before we understand what they were. This is why the recent expeditions matter. Why the 100 new species in 1 month matters. We're racing against time, racing to document biodiversity before it disappears. Every expedition finds something new. Every new species is a victory. A species saved from anonymous extinction. Given a name, given a description, given a chance to be remembered. But we can't discover everything fast enough. The ocean is too big, too deep, too expensive to explore. We're doing our best, but it's not enough. That's the reality. We're trying. Scientists are working. Funding is limited. Technology is improving. ROVs are better, cheaper, more capable. But there's still so much we don't know, so much we'll never know. The ocean keeps its secrets. Life in the deep continues, adapting, surviving, thriving, whether we watch or not, whether we understand or not, whether we care or not. The blobfish floats in the darkness. The vampire squid eats marine snow. The anglerfish waits for prey. The barrelly stares upward through its transparent head. The giant squid hunts in the midnight zone. And we surface, dwellers remain mostly ignorant, looking at the ocean from above, thinking we understand, thinking we know. but we don't know. We've seen fragments, glimpses, a few hours of ROV footage from a few locations, a few specimens in museums, a few papers published. It's not enough. It's never enough. The ocean is 99% unexplored, unmapped, unknown. We assume it's mostly empty, mostly boring, mostly the same everywhere. But every time we look, we find diversity, complexity, weirdness, life that shouldn't work but does, fish with see-through heads, squid from hell, males that fuse to females, creatures walking on fins, urchins like cacti, worms that eat bones, corals that live for millennia, and we're just scratching the surface. The deep sea holds answers. Questions we haven't thought to ask. Medical compounds, new biochemistry, novel adaptations, solutions to problems we have on land. How do deep-sea creatures handle pressure? Could we apply that to submarine design? How do they survive low oxygen? Could that help us understand hypoxia? How do they live so long? Could that inform aging research? How do their proteins fold under pressure? Could that improve our protein engineering? The deep sea is a library, full of information, written in DNA, in biochemistry, in morphology. But we can't read it if we destroy it, can't learn from it if it's gone, can't apply it if we never discover it. This is why we explore, why we fund research, why we protect the ocean, not just for the ocean's sake, for ours. We need the ocean. We depend on it. Oxygen. Half of Earth's oxygen comes from marine phytoplankton, climate regulation. The ocean absorbs heat, absorbs carbon dioxide. Food. 3 billion people depend on the ocean for protein, jobs, fishing, shipping, tourism. The ocean economy is worth trillions. We can't afford to lose it, we can't afford to ignore it, we can't afford to destroy it, but that's what we're doing. Slowly, surely, thoughtlessly. The good news is we can stop, we can change, we can protect. We have the technology, the knowledge, the resources. We just need the will, the political will, the social will, the individual will. Make better choices. Eat sustainable seafood. Reduce plastic use. Support ocean conservation. Vote for climate action. Demand corporate responsibility. Make noise. Spread awareness. Share information. Educate others. The ocean needs advocates, champions, protectors. It needs us, all of us. In November 2025, researchers from the Senckenberg Ocean Species Alliance are still analyzing samples from recent expeditions, genetic work, morphological studies, taxonomic descriptions. Every sample could be a new species. Every analysis could reveal something unexpected. They're using advanced methods, DNA barcoding, CT scanning, 3D reconstruction. It's faster than old methods, more accurate. But still slow, still expensive, still limited by funding and personnel. There aren't enough taxonomists, not enough deep-sea biologists, not enough funding for exploration. We need more, much more. The ocean can't wait. The Schmidt Ocean Institute continues expeditions, the Falkor vessel, the ROV Sebastian. They're mapping seamounts, discovering species, sharing data openly, making it available to researchers worldwide. That's crucial. Open access, open data. Everyone can use it. Everyone can learn. Everyone can contribute. Science works best when it's collaborative, when information flows freely, when resources are shared. The ocean belongs to everyone. The data should too. The Monterey Bay Aquarium Research Institute continues ROV dives, documenting deep sea life, creating video archives, publishing research. They've been doing this for decades. They have the longest continuous record of deep sea observations anywhere. Their data shows changes over time, species distributions shifting, oxygen levels declining, temperature rising. It's a warning, a record of human impact. We're changing the deep sea, faster than we thought, more than we expected, and we're just beginning to measure it. Ocean Census is coordinating global efforts, connecting researchers, funding expeditions, supporting taxonomy. Their goal is to discover marine species faster, Document them before they disappear. It's ambitious, it's necessary, it's not enough, but it's something. They're making progress. Hundreds of species described, thousands more in the pipeline, but millions remain unknown. The gap is huge. Closing it requires unprecedented effort. Global cooperation, massive funding, decades of work, maybe centuries. But we have to try. Universities worldwide are training new ocean scientists, marine biologists, deep-sea ecologists, taxonomists, engineers. The next generation, they're learning new techniques, using new technology, thinking new thoughts. They'll continue the work, make new discoveries, push boundaries. We're counting on them. The ocean is counting on them. The future depends on them. And on us, on what we do now, on what we choose to protect, on what we decide matters. The ocean is not empty. The deep is not lifeless. The abyss is not boring. It's full, diverse, strange, wonderful, terrifying, beautiful. It's the largest ecosystem on Earth, the oldest, the least known, and we're running out of time to understand it. Every species we discover is a gift. Every ecosystem we protect is a victory. Every choice we make matters. The blobfish, the vampire squid, the anglerfish, the barrelay, the giant squid. They're all connected to each other, to us, to the planet. Protecting them protects us, destroying them destroys us. It's that simple, that complicated, that urgent. So what do? We learn, we share, we care, we act. We explore the ocean. We map the seafloor. We discover the species. We describe them. We name them. We study them. We understand them. We protect them. We create marine reserves. We ban deep-sea mining. We regulate fishing. We reduce emissions. We clean up pollution. We change. We adapt. We survive. Together, with the ocean. Not despite it, not against it. With it, part of it, dependent on it, responsible for it. That's the only way forward. The only way to ensure there's a future for the deep sea, for us, for everything that depends on the ocean, which is everything, everyone, all of us. The blobfish floating in the darkness, the vampire squid eating marine snow, the anglerfish with its glowing lure, the barreleye staring through its transparent head, the giant squid hunting with dinner plate eyes, and the hundreds of species we just discovered, and the millions we haven't found yet. They're all out there, right now, living, existing, adapting, surviving, in the cold, the dark, the pressure, the deep. Our ocean, our planet, our responsibility, our choice. What do we choose? What do we protect? What do we destroy? The answer is simple. The execution is hard, but we have to try. The ocean has no voice. We have to speak for it, loudly, constantly, urgently, now, before it's too late, before the species disappear, before the ecosystems collapse, before we lose something we can never replace, something we never fully understood, something that existed for millions of years, something we destroyed in decades. That's the future if we don't act. That's the reality if we don't care. That's what happens if we stay silent. So don't stay silent. Speak up. Learn more. Share more. Care more. Act more. The ocean is waiting. The deep is watching. The creatures are depending on us. On you. On me. On everyone who hears this. Everyone who cares. Everyone who wants a future where the blobfish still floats. Where the vampire squid still drifts. Where the anglerfish still hunts. Where the barreley still stares. Where the giant squid still swims. Where the ocean still thrives. That's the goal. That's the mission. That's what we fight for. Starting now. Starting here. Starting with one choice. One action. One voice. Yours. In January 2024, in the waters off Chile, scientists found 100 new species in less than 30 days. They didn't expect that many. They didn't think it was possible, but it was. Because the ocean is full of surprises, full of life, full of things we've never seen, things waiting to be discovered, things that have existed forever but we just didn't know, didn't look, didn't care enough to find them. Until now. Until we sent the ROV down. Until we pointed the cameras. Until we collected the samples. Until we looked, really looked, and saw, really saw. Not just water, not just darkness, not just empty ocean, but life. weird life, strange life, impossible life, life that redefines what we thought we knew, life that challenges our assumptions, life that exists despite the odds, because of the odds, life that adapted, survived, thrived, in conditions we can barely imagine, conditions that would kill us instantly, conditions that seem hostile, impossible, uninhabitable, but aren't. Because life finds a way, always has, always will, in the deep, in the cold, in the dark, in the pressure, life persists, and we're just beginning to understand how, just beginning to see why, just beginning to realize what we're risking when we threaten these ecosystems, what we're losing when species go extinct, what we're destroying when we trawl the seafloor, what we're ignoring when we focus only on land, only on what we can see from the surface, only on what seems to matter. But it all matters. Every species, every ecosystem, every seamount, every vent, every seep, every deep sea fish with transparent head, every squid eating garbage, every male fused to female, every adaptation that seems bizarre but works perfectly in context. It all matters. It's all connected. It's all part of the same system, the same planet, the same ocean, the same future. Our future, depending on choices we make now, actions we take today, priorities we set tomorrow. The ocean can't wait, the species can't wait, the deep sea can't wait. Time is running out, species are disappearing, ecosystems are collapsing. And we're still exploring, still discovering, still learning, still racing against extinction, against ignorance, against apathy, racing to understand before it's gone, before it's too late, before the last blobfish dies, before the last vampire squid fades away, before the last anglerfish stops glowing, before the deep goes dark. Forever. That's what's at stake. That's what we're fighting for. That's why this matters. Why you matter. Why your voice matters. Why your choices matter. The ocean needs you, the deep needs you, the weird, strange, impossible creatures need you, to learn about them, to care about them, to protect them, to speak for them. Because they can't speak, can't vote, can't change policies, can't save themselves from what we're doing, what we're still doing, what we'll keep doing, unless we stop, unless we change, unless we choose different, choose better, choose ocean, choose life, choose future, choose now, not later, now, today, this moment, this choice. What will you choose? Let's go deeper into the specific discoveries that changed everything. The year 2004 marked a turning point, not just for the barreli, for everything. Mbari's ROV technology reached maturity. They could film at depths previously impossible. Stay down longer, move more precisely, see more clearly. The footage started accumulating. Hours became days, days became weeks, years of observations, and patterns emerged. Behaviors nobody predicted, species interactions nobody imagined. The deep wasn't the desert everyone thought. It was a complex web, interconnected, dynamic, alive in ways that surprised everyone. Take the glass sponges again. Scientists knew they existed, knew they were old, but they didn't know about the electrical signals. In 2006, researchers discovered that glass sponges conduct electricity through their silica skeletons. Not like nerves, different, slower, but functional. The sponge can sense when something touches it, can respond, close its pores, stop filtering, wait for the threat to pass, then reopen, resume feeding. A sponge, with no brain, no neurons, no muscles, but it can sense and respond. How? Through the glass structure itself. The silica conducts signals. Researchers are still figuring out the mechanism, still trying to understand how an animal with no nervous system can coordinate activity across its entire body. It challenges everything we know about sensory systems, about what's required for sensation, about the minimum complexity needed for awareness. Maybe awareness is the wrong word. Maybe response is better. Maybe the sponge doesn't feel anything, just reacts, mechanically, chemically. But even that is remarkable. Even that is something we didn't expect. The Pompeii worm, Alvinella pompeiana. lives at hydrothermal vents, builds tubes, sticks the tail end into the vent, where water reaches 80 degrees Celsius, 176 Fahrenheit. The head end stays in water that's 20 degrees, 68 Fahrenheit, 60-degree temperature difference, across 10 centimeters, 4 inches. The worm's body handles this gradient. The proteins at the tail end are heat-stable, modified, different from the proteins at the head end. The worm is biochemically asymmetric, different chemistry in different body parts, adapted to occupy an impossible niche, the hottest temperature any animal tolerates, the steepest thermal gradient any organism survives, and the worm thrives there, reproduces there, lives its entire life there. It has symbiotic bacteria coating its back. The bacteria form a fleece, insulation. They also produce nutrients, chemosynthesis. The worm gets food from the bacteria. The bacteria get hydrogen sulfide from the vent water. Mutualism, partnership, survival through cooperation. The Yeti crab, Kiwa hirsuta, discovered in 2005, South Pacific. Hydrothermal vents, 2,300 meters deep, 7,500 feet. It's white, furry. The furry appearance comes from setae, hair-like structures covered in bacteria. The crab farms the bacteria, waves its claws in the vent water, exposes the bacteria to hydrogen sulfide. The bacteria grow. The crab eats the bacteria, scrapes them off its claws with specialized mouthparts. It's cultivating food, farming, underwater bacterial agriculture. The crab is blind, eyes reduced to almost nothing, just light-sensitive patches. It doesn't need vision, it needs the bacteria. It positions itself near vents, waves its claws, harvests, repeats. Simple life, effective life. Since 2005, multiple Yeti crab species have been discovered. Kiwa puravida in Costa Rica, Kiwa tyleri near Antarctica. Different vents, different species, but same strategy, bacterial farming, convergent evolution across the globe. The deep sea selects for what works, and bacterial farming works at hydrothermal vents. Multiple lineages discovered it independently. Crabs, shrimp, snails. They all farm bacteria. They all use chemosynthesis indirectly. They can't do it themselves, but they cultivate organisms that can. It's sophisticated, it's elegant, it's bizarre. The pelican eel. Uripharynx pelecanoids. Mouth larger than its body. Jaws that unhinge completely. Stomach that inflates like a balloon. Found at depths of 500 to 3,000 meters. 1,600 to 9,800 feet. Length up to 75 centimeters, 30 inches, but most of that is tail. Thin whip-like tail. The body is small. The mouth is huge, disproportionate. It can swallow prey larger than itself. Fish, squid, crustaceans, just opens wide, engulfs, swallows. The stomach expands to accommodate. The eel probably doesn't eat often. Can't. Food is scarce. But when food appears, it can take advantage. Can eat big meals, store energy, survive long fasts. The tail has a light-producing organ, pink bioluminescence, function unknown. Maybe attracts prey, maybe attracts mates, maybe both. The eel was first described in 1882. We've known about it for over a century, but we've barely seen it alive. A few photographs, a few video clips. That's it. Most specimens are dead, caught in nets, examined in museums. We don't know how it lives, how it hunts, how it reproduces. Basic biology remains mysterious. The coffin fish, Chonacidi, related to anglerfish, but different body plan. Box-shaped, compressed, covered in spines. Small fins. They sit on the seafloor. Don't swim much. Wait for prey. They have a lure, like anglerfish. But it's retractable, withdrawn into a groove on the head when not in use, extended when hunting. The mouth is huge, opens wide, suction feeds, water rushes in, prey follows, mouth snaps shut, over. Some species are bright red, some are pink, some are brown. They're found worldwide, depths of 50 to 2,000 meters, 160 to 6,500 feet, multiple species, at least 24 described, probably more exist. They're small, 10 to 30 centimeters, 4 to 12 inches. Easy to overlook, easy to miss in trawl samples. Each species slightly different. Different spines, different colors, different depth ranges. We're still sorting them out, still describing new ones, still learning which features matter for taxonomy. The black swallower, Chiasmodon niger, small fish, big appetite, length 25 centimeters, 10 inches. It can eat fish twice its length, 4 times its mass. The stomach is distensible, stretches enormously. The jaw can unhinge, open wide. The fish attacks prey larger than itself, bites, holds on, gradually works the prey into its mouth, swallows. The stomach expands, sometimes too much. Sometimes the prey is so large it starts decomposing before the swallower can digest it. Gases build up. The swallower floats to the surface, dies, washes ashore. That's how scientists usually find them, dead, bloated, with enormous prey still in the stomach, visible through the distended belly wall. It's gruesome, it's fascinating. It's a strategy that sometimes fails catastrophically, but often enough it works. The species survives, reproduces, continues. The stoplight loosejaw, Malacosteus niger, a dragonfish. With a unique adaptation, it produces red bioluminescence. Most bioluminescence is blue or green. Red is rare. Most deep-sea creatures can't see red, but the stoplight loosejaw can. It has a red, sensitive pigment in its eyes, derived from chlorophyll. Bacteria in its diet provide the chlorophyll. The fish modifies it, incorporates it into visual pigments. Now it can see red light, its own red light. It shines red light on prey. The prey doesn't see it, doesn't react. The loosejaw sees it clearly. Invisible spotlight. It hunts with an advantage. Sees prey that thinks it's hidden. The jaw is loosely attached, barely connected to the skull. Opens extremely wide, catches prey. The teeth are long, transparent, made of organic materials, not mineralized. They're flexible, curved, point backward. Prey can enter, can't escape. The loosejaw swallows, continues hunting. The red light strategy is unique. Only a few species have it. Only in the Malacosteus genus. It evolved once. Stayed rare, but effective. Lethally effective. The fangtooth, Anoplogaster cornuta. Teeth so large they don't fit in the mouth. The longest teeth relative to body size of any fish. The fangs are up to 6 centimeters long, 2.4 inches. The fish itself is only 15 to 18 centimeters, 6 to 7 inches. The teeth are huge, disproportionate. When the mouth closes, the teeth slide into special pockets in the skull. Otherwise they'd pierce the brain. It's an extreme adaptation. For catching prey. For holding prey. Nothing escapes those fangs. The fangtooth lives at depths of 200 to 2,000 meters, 650 to 6,500 feet. Juveniles live shallower, adults deeper. They undergo vertical migration, not daily, ontogenetic. As they age, they descend. The young feed on crustaceans. The adults eat fish, small lanternfish, hatchetfish, anything they can catch. They hunt by ambush, sit and wait, snap, hold, swallow. They're ugly, probably the ugliest fish in the sea. Small eyes, large head, huge fangs, dark brown or black coloration. They look like nightmares, but they're just fish. Doing what fish do, eating, surviving, reproducing, same as everything else, just with bigger teeth. The telescope octopus, Amphitrechus pelagicus, transparent body, tubular eyes, like the barrel eye, but it's an octopus, lives in the water column, pelagic, not benthic, depths of 500 to 3,000 meters, 1,600 to 9,800 feet, small, 8 centimeters mantle length, 3 inches. The eyes are large, protrude from the head, point forward, high visual acuity. The body is nearly invisible, gelatinous, transparent. You can see internal organs, the digestive system, the gills. It's a floating octopus made of glass. Why transparent? Camouflage. Predators can't see it. Prey can't see it. It drifts through the water, nearly invisible, sees everything with those large eyes. It probably eats small crustaceans, amphipods, copepods, larvae, catches them with the arms, brings them to the beak. We don't know much more. Few specimens have been collected, few observations made. It's another mystery. Another creature we know exists but barely understand. The Dana octopod, Grimpoteuthis bathinectes, another Dumbo octopus, but larger, up to 35 centimeters, 14 inches, lives at depths of 4,000 to 5,000 meters, 13,000 to 16,000 feet. Among the deepest dwelling octopuses, it has the characteristic ear-like fins, flaps them to swim, hovers above the seafloor. The body is gelatinous, soft. The mantle is short, the arms are long, connected by webbing. When it spreads the web, it looks like an umbrella. Parachute. It descends slowly, arms spread, web extended, lands on the seafloor, covers an area, traps anything underneath. Snails, worms, small crustaceans. The arms converge, bring food to the mouth, simple, effective. The octopod is pink or purple. Color varies, eyes are small, vision probably limited. It relies more on touch, chemical sensing. The suckers on the arms detect texture, taste. Everything that touches them provides information. The octopod processes this, decides what's food, what's not, eats accordingly. The cirrate octopuses, the group that includes Dumbos, they're different from other octopuses. They have cirri, finger-like projections between the suckers. Function unclear. Maybe sensory, maybe help with gripping. They have internal cartilaginous supports, primitive shells, remnants of ancestral shells that other octopuses lost completely. They have fins. Other octopuses use jet propulsion. Cirrates use fins. Different locomotion, different lifestyle. They're deep-sea specialists, all of them. No cirrate octopus lives in shallow water. They diverged from other octopuses millions of years ago, took a different path, went deep, stayed deep. Now there are about 50 species, all in the deep, all with fins, all gelatinous, all weird. The cookie-cutter shark returns, Icystius brasiliensis. Let's talk more about it. The bioluminescence, the belly glows, counter-illumination, matches the light from above. But the collar around the throat doesn't glow. Dark patch. From below, it looks like a small fish, silhouette of prey. Larger predators approach, try to eat the small fish. The cookie cutter attacks, bites, removes flesh, escapes. It's using itself as bait. The dark collar is a lure, predatory mimicry. The shark tricks predators, makes them come close, then becomes the predator. It's brilliant, it's deceptive. It's a strategy that shouldn't work, but does. The shark feeds on tuna, dolphins, whales, seals, even great white sharks. Wounds have been found on great whites, cookie-shaped bites. The biggest predator in the ocean gets bitten by a small, deep sea shark. The ocean is full of these inversions, these subversions. Size doesn't guarantee safety. Strength doesn't prevent attack. The deep has specialists, niche predators, species that exploit weaknesses. The cookie cutter found a weakness. Large predators are curious. They investigate potential prey. The cookie cutter exploits that curiosity, bites them, survives because it's fast, gone before they react. It's hit and run, guerrilla warfare, survival through deception and speed. The megamouth shark, Megachasma pelagios, discovered in 1976 off Hawaii, caught accidentally by a research vessel. Nobody knew it existed. A 5-meter shark, 16 feet, living in moderately deep water, and we'd never seen one. It's a filter feeder, like whale sharks and basking sharks, but different, smaller mouth, different feeding mechanism. It swims with mouth open, water flows through. Gill rakers trap plankton, krill, small jellyfish. The mouth is bioluminescent, glows white. Maybe attracts prey, maybe just reflects light. Function debated. The shark performs daily vertical migration, deep during day, shallower at night, follows the deep scattering layer, the massive aggregation of zooplankton and small fish that rises at night, falls at dawn. The The megamouth follows, feeds, returns to depth. Since 1976, only about 120 megamouth sharks have been seen. That's 120 sightings in nearly 50 years, 3 per year globally. It's one of the rarest sharks, one of the largest species we barely know. Where do they reproduce? How? Often, what's the population size? We don't know, we have guesses, estimates, but not data, not enough data. The megamouth remains a ghost, present but barely seen. Each new sighting makes news. Each specimen teaches us something. But progress is slow, very slow. The frilled shark returns. Let's examine reproduction more. Ovoviviparous, internal development. But the eggs don't hatch immediately. They remain in the female, developing. For 3.5 years, the longest gestation of any vertebrate, longer than elephants, 22 months, longer than alpine salamanders, 3 years, the frilled shark beats them all, 3.5 years. Why? We think it's the cold. Low temperatures, slow development. Everything takes longer in the deep. Growth, maturation, reproduction. The embryos develop slowly, very slowly. The female carries 2 to 15 embryos, usually around 6. Large eggs, yolk sacs. The embryos absorb the yolk. Grow slowly. Finally, after 3.5 years, they're ready. Born live, fully formed, about 40 to 60 centimeters, 16 to 24 inches. Ready to hunt immediately. No parental care. The young are independent from birth. They sink to appropriate depth, start hunting, start surviving. The frilled shark reaches sexual maturity late. Males at 1.5 meters, 5 feet. Females at 1.8 meters, 6 feet, age unknown. Probably 10 to 15 years, maybe longer. Then they can reproduce. Long maturity, long gestation, low fecundity. Classic K selection. It makes the species vulnerable. Fishing pressure could collapse populations quickly. Recovery would take decades, but frilled sharks aren't targeted commercially. Caught as bycatch occasionally, not often. Populations seem stable, for now. But climate change could affect them. Warming water, changing currents, declining oxygen. The frilled shark is adapted to cold, stable conditions. Change those conditions, and it might struggle. Now examine bioluminescence more deeply. Why is it so common in the deep? Several reasons. Counter-illumination, we've discussed this. Match the light from above, become invisible from below. It works. Multiple families evolved it independently. Hatchetfish, lanternfish, some squid, some shrimp. Widespread strategy, luring. Anglerfish are the obvious example, but others do it too. Dragonfish, cookie-cutter sharks, loose jaws, produce light, attract prey, catch prey. Simple, effective. Communication, finding mates in the dark. Bioluminescence helps. Flash patterns, species-specific. Males and females recognize each other, meet, reproduce. Without light, meeting would be nearly impossible. Defense, the vampire squid's bioluminescent mucus, Atola jellyfish. They flash when attacked. Create light show, confuse predators, attract secondary predators. Burglar alarm. Make the predator visible, make it vulnerable. Often the predator flees, releases the prey. Both survive. It's a last resort. Costs energy, but it works often enough. How does bioluminescence work? Luciferin, a light-producing molecule, luciferase, an enzyme. They react, produce light. Energy released as photons. Very efficient, almost no heat. Nearly all energy becomes light. 90% efficiency. Compared to incandescent bulbs, 5% efficiency, 95% wasted as heat. Bioluminescence is vastly more efficient. Different organisms use different luciferins, different luciferases. Convergent evolution, same function. Different chemistry. Some organisms make their own luciferin, others get it from diet. Some harbor symbiotic bacteria. The bacteria produce light. The host provides habitat, food, protection. The bacteria provide light, partnership. The anglerfish's estuary is full of bacteria. The flashlight fish has bacteria in organs under its eyes, can cover them with a shutter, flashes light on and off. The bacteria glow constantly. The fish controls visibility. smart, efficient. Bioluminescence colors range from blue to red. Blue is most common, travels farthest in water. Red and yellow are absorbed quickly. Don't travel far. Blue penetrates. That's why most deep sea bioluminescence is blue. It works. It communicates across distance. The exceptions are interesting. Red bioluminescence in loose jaws. Hardly anyone can see it. That's the point. Private channel. See prey without being seen. Green bioluminescence in some jellies. Some squid still visible. Still travels reasonably far, but different from blue. Maybe species recognition. Maybe niche separation. Maybe historical accident. We don't always know why. The distribution of bioluminescence is revealing. Rare in surface waters. Common in twilight zone. Very common in midnight zone. Ubiquitous in the abyss. As depth increases, bioluminescence increases. Why? Because it's dark. In shallow water, sunlight provides illumination. Bioluminescence can't compete. But remove sunlight and bioluminescence becomes valuable, becomes necessary. Nearly every deep-sea species has bioluminescence or lives with organisms that do. It's not optional. It's essential. Light in the darkness. Communication in the void. It's how the deep sea works. It's how life persists where no light should be. Scientists estimate that 76% of pelagic animals in the ocean are bioluminescent. 3/4. That's the majority of ocean life by number. Not by mass. Most are small. Copepods, krill, small fish, small squid. But by abundance, bioluminescent animals dominate. The ocean glows at night, in the depths. It's full of living light, flashes, steady glows, trails, clouds. The darkness isn't empty, it's alive with light. Just not sunlight. Living light, biological light, light that evolved, light that serves purpose, light that matters. Consider the ostracods, Vargula hilgendorfii, small crustaceans, few millimeters long. They produce bioluminescent displays. Males court females with light. They release glowing clouds, puffs of luciferin and luciferase. The chemicals mix in seawater, glow. Each species has a unique pattern, different rhythm, different brightness, different duration. Females watch, choose, select males based on display quality. It's sexual selection, driven by bioluminescence. The male with the best display gets to mate. Poor displays get ignored. It's competition, light-based competition. In the ocean, dinoflagellates, single-celled algae, many species bioluminescence. When disturbed, they flash. Blue light, brief. This is what causes bioluminescent bays, bioluminescent waves. Water full of dinoflagellates. Movement triggers flashing. Waves glow. Boats leave glowing wakes. Hands trail light. It's beautiful. It's famous. Tourist attractions in Puerto Rico, California, Australia. Other locations. But why do dinoflagellates flash? Defense. When something disturbs them, they flash. The flash makes the disturbance visible. Small crustaceans grazing on dinoflagellates become visible when they trigger flashing. Fish see the glowing crustaceans, eat them. The dinoflagellates survive. Burglar alarm again. Make the predator visible. Recruit larger predators. Increase survival. It's sophisticated for single cells. Jellyfish bioluminescence. Many species have it. Equaria victoria, crystal jellyfish, Pacific coast. They have green fluorescent protein. GFP. This protein revolutionized biology. Scientists extracted it, learned to express it in other organisms. Now GFP is used as a marker in research worldwide. Thousands of studies, Nobel Prize awarded, all because of a glowing jellyfish. The jellyfish uses GFP to convert blue bioluminescence to green, more visible, more effective. The jellyfish doesn't know it changed science, doesn't know we use its protein, doesn't care, it just glows, just exists. But that existence mattered. Changed human knowledge, changed medical research, changed everything. That's the thing about the ocean. About deep-sea creatures, they exist, they adapt, they survive, and sometimes randomly they change human civilization. Velcro inspired by burrs, but burrs are land. Shark skin inspired swimsuits. Dolphin skin inspired ship coatings. Hagfish slime studied for body armor. Spider crab shells for wound dressings. The ocean is full of solutions. Biomimicry, learning from nature, applying it. The deep sea is particularly rich. Extreme adaptations, novel solutions, things that don't exist on land. Pressure-resistant proteins, antifreeze compounds, unique enzymes, bioluminescent systems, all potential applications, all waiting to be discovered, studied, applied, but only if they still exist. Only if we don't destroy them first. The Antarctic toothfish, Disosticus morsoni, lives in freezing water, -1.9 Celsius, 28.6 Fahrenheit, below the freezing point of fresh water. But seawater freezes lower. The toothfish survives, has antifreeze proteins in its blood, glycoproteins. They bind to ice crystals, prevent growth, ice can't form. Blood stays liquid. The fish survives. These antifreeze proteins are studied. Potential applications, food preservation, organ transplantation, anything needing protection from ice. The toothfish doesn't know it's useful. It's just surviving. Using chemistry that evolved over millions of years, chemistry we're now learning to use. The same applies to pressure-resistant proteins. Deep-sea creatures have proteins that function at 1,000 atmospheres. Our proteins denature under pressure, unfold, stop working. Deep-sea proteins don't. They're structured differently, stabilized differently. Scientists want to understand how, want to engineer pressure-resistant proteins, industrial applications, extreme environment applications, space applications maybe. The deep sea shows it's possible, provides examples, natural, experiments, millions of years of evolution testing solutions. We just have to learn, have to study, have to preserve the organisms long enough to understand them. Back to creatures, the chimeras, ratfish, ghost sharks, order chimeriforms, related to sharks and rays but different, split off 380 million years ago, ancient lineage. They have one gill opening. Sharks have 5 or 6. They have a venomous spine in the dorsal fin. They have a clasper on the head, males only, used in mating. How? Unknown. They're found worldwide. Deep water usually, some species to 2,500 meters, 8,000 feet. They eat shellfish, crustaceans, echinoderms, crush them with plate-like teeth, not individual teeth, tooth plates, grind shells, powerful jaws. They're caught as bycatch, sometimes targeted for meat, liver oil, populations declining in some areas. They grow slowly, mature late, low reproduction, vulnerable to overfishing. The snipe eel, Nemicthyidae, extremely long jaws, curved, don't meet, can't close completely, lined with tiny teeth, backward pointing. The eel sweeps its jaws through the water, catches small crustaceans, trapped on the teeth, swallowed. It's a specialized feeding method, works only for small prey, but small prey is abundant. Copepods, amphipods, everywhere. In the ocean, the snipe eel is efficient at catching them, length up to 1.3 meters, 4.3 feet, but very thin, ribbon-like. The jaws account for significant length. The body is slender, almost fragile-looking, found at depths of 300 to 2,000 meters, 1,000 to 6,500 feet, distributed worldwide. Multiple species, 15 to 20 depending on taxonomy. They're caught occasionally, studied occasionally. Basic biology known, details lacking. Ribbonfish, Trachipteridae, not the same as snipe eels. Different fish, long, thin, ribbon-shaped, silver, red fins. They swim vertically, head up, body hanging down, undulate through the water. It looks strange, unusual orientation, but it works. They're found worldwide. Some species reach 8 meters, 26 feet. Giant ribbons. They live at depths of 20 to 1,000 meters, 65 to 3,300 feet. occasionally wash ashore dead, injured. These strandings create legends, sea serpent stories, oarfish, messengers from the sea god, harbingers of earthquakes. Japan has folklore about them. Reality is simpler. They're fish. Dying, coming ashore, no magic, no portent, just biology. The gulper eel again. Eurypharynx pelechinoids. Let's examine the tail more closely. Long, whip-like, about 60% of total length. Thin, fragile-looking. At the end, a light organ. Photophore, pink light, complex organ. Multiple chambers, can flash or glow steady, can vary intensity, probably used for attracting prey, maybe attracting mates. The eel swims with undulating movements of the tail. The light draws attention. Small fish approach, investigate. The eel's huge mouth is nearby, waiting, mouth opens, suction, prey engulfed, over. The tail might also stabilize swimming, act as a rudder, balance the huge mouth. The eel's body plan is extreme. All mouth and tail, minimal body. It works. The species survives. Multiple specimens collected. Distribution worldwide. Temperate and tropical oceans. 3 species in the family, each slightly different, each occupying slightly different depths. Niche partitioning. The spookfish, Dolichopteryx longips, a barrelly relative, but stranger. It has 4 eyes, sort of. Each eye is divided. Upper part looks up, has a lens, normal eye structure. Lower part looks down, no lens, uses a mirror. Crystalline reflective plates. Focus light. It's the only vertebrate with mirror-based eyes. Other animals use mirrors. Scallops, some crustaceans. But in the vertebrates, only the spookfish. Why? It solves a problem. Looking down in the deep. Below, there's almost no light. A lens can't gather enough. A mirror can. Collects light across a wider area. Focuses it. The spookfish sees down, sees bioluminescence from below, sees prey, sees predators. It's an evolutionary innovation. Unique solution. Works only for this species, this niche, this lifestyle. Evolution found it, tested it, kept it. The spookfish lives at depths of 400 to 2,500 meters, 1,300 to 8,200 feet. Pacific Ocean. Some parts of Atlantic and Indian Oceans. Rare. Difficult to catch. Few specimens in collections. The mirror eyes were discovered in 2009. Researchers from Germany and UK published in Current Biology. They used electron microscopy, imaging techniques, found the crystalline plates, realized they form a mirror, calculated the optics, showed it works. It was revolutionary. A new type of vertebrate eye. Nobody expected that. Nobody predicted mirrors, but there it is. The ocean invents, innovates, solves problems we haven't thought of, uses methods we don't predict, keeps surprising us, will always surprise us. Because we're land animals, we think in land terms. The ocean thinks different, lives different, is different. But we're running out of time, discovering species, describing them, publishing papers, but also destroying habitat, extracting resources, changing climate. The race is unequal. Discovery proceeds slowly. Destruction proceeds quickly. We're losing species we haven't named, ecosystems we haven't mapped, resources we haven't identified. It's a catastrophe unfolding, silently, in the dark, where nobody sees, nobody except scientists, and they're screaming, writing papers, giving talks, testifying. Nobody listens. Industry wants profit, politicians want donations, public wants cheap goods. The ocean pays the price, life pays the price, future generations pay the price. But right now, today, the species still exist. The ecosystems still function. The ocean still produces oxygen, still regulates climate, still provides food. We can still protect it, still save it, still choose different, better, sustainable. If we act now, not later, now, before it's too late, before the last population collapses, before the last ecosystem fails, before the ocean dies, bit by bit, species by species, until there's nothing left, except jellyfish, bacteria, tough generalists, survivors, but not the diversity, not the complexity, not the incredible adaptations, not the blobfish, not the vampire squid, not the anglerfish, not the barreleye, not any of the weird, wonderful, impossible creatures that make the deep sea fascinating. They'll be gone, forever. And we'll have been the cause. The generation that destroyed the ocean, that ended 3 billion years of marine evolution, that decided profit mattered more than life, that chose convenience over survival, that looked at the wonder of the deep and decided it wasn't worth saving. That's who we'll be. Unless we change, unless we fight, unless we care enough to act, to vote differently, to buy differently, to live differently, to prioritize the ocean, the planet, the future, over short-term gain, over convenience, over apathy. The choice is ours, the time is now, the creatures are waiting. Depending on us, on you, on everyone who cares, who understands, who refuses to let this happen. The ocean needs champions. Be one, today, now, this moment, because tomorrow might be too late. For the blobfish floating in darkness, for the vampire squid eating marine snow, for the anglerfish glowing in the abyss, for all of them, for all of us, for the ocean that gives us everything and asks only that we don't destroy it. That's not too much. That's the bare minimum. Can we do it? Will we do it? The answer determines everything. Everything that comes next. Everything that survives. Everything that matters. What's your answer?